Assalamu alaikum. My name is Umayra. I'm from chemistry department. Today the topic of my presentation is stereochemistry of carbohydrates. First of all, I'll discuss stereochemistry in point of view of Fisher projections because carbohydrates usually have numerous chirality centers. It was recognized long ago that a quick method for representing their stereochemistry was needed. So, chirality center is actually a tetrahedral atom in a molecule which is bearing four different ligands with lone pairs. In 1891, the German chemist Emil Fischer suggested a method based on the projection of tetrahedral carbon atom onto a flat surface. These Fischer projections were soon adopted and are now a common means of representing stereochemistry at chirality centers, particularly in carbohydrate chemistry. A tetrahedral carbon atom is represented in a Fischer projection by two crossed lines. The horizontal lines represent bonds coming out of the page, and the vertical lines represents bonds going into the page. Like into the below figure, horizontal lines represent bonds coming out of the page, and the vertical lines represent bonds going into the page. Moving towards the simplest monosaccharide can be drawn as like this. In this example, the structure of glyceraldehyde is drawn in light of the Fischer projections. So because a given chiral molecule can be drawn in many ways, but it's sometimes necessary to compare two projections to see if they represent the same or different enantiomers. To test for identity, Fischer projections can be moved around on the paper, but only in two kind of motions. Moving a Fischer projection in any other way inverts its meaning. So we see what are two kinds of motions which are allowed. A Fischer projection can be rotated on the page by 180 degree but not by 90 degree or 270 degree. Only a 180 degree rotation maintains the Fischer convention by keeping the same substituent groups going into and coming out of the plane. In the following Fischer projection, like this in this glyceraldehyde, for example, the hydrogen and hydroxyl groups coming out of the plane both before and after a 180 degree rotation. Now the question is why rotation a 90 degree is not allowed? The reason is at 90 degree rotation breaks the Fischer convention by exchanging the groups that go into the plane with those that come out. But in the following Fischer projection of glyceraldehyde, the hydrogen and hydroxyl groups originally come out of the plane like in this figure, but go into plane after a 90 degree rotation. A Fischer projection can also have one group held steady while the other three rotate in either a clockwise or counterclockwise rotation. The effect is simply to rotate around a single bond which doesn't change the stereochemistry. RS stereochemical designations can be assigned to the chirality centers in a Fischer projection by following three steps. Step 1. Rank the four substituents in usual way. Step 2. Place the group of the lowest ranking, usually hydrogen, at the top of the Fischer projection by using one of the allowed motions. This means that the lowest ranked group is oriented back, away from the viewer as required for assigning configuration. Step number 3. Determine the direction of rotation 1 and 2 and 3 of the remaining three groups and assign R or S configuration. Carbohydrates with more than one chirality center are shown in feature projections by striking the centers on the top of one another with a carbonyl carbon on or the near the top. Glucose, for example, has four chirality centers stacked on the top of one another in a Fischer projection. Such representations do not. However, an accurate picture of the molecule's to three-dimensional conformation, which is circled around itself like a bracelet, like in this figure, which is actual Fischer projection. Then we discussed DL sugars. What are DL sugars in the carbohydrates? Glyceraldehyde, the simplest aldose, one of the monosaccharides, has only one chirality center and thus has two enantiomeric forms, which are non-identical mirror image forms actually. Only the dextrorotatory enantiomers occurs naturally. However, that is a sample of naturally occurring glyceraldehyde placed in a polarimeter. It is plain polarized light in a clockwise direction denoted one. Since one glyceraldehyde has been found to have an R configuration at carbon number 2, it can be represented by official projection for historical reasons dating back long before the adoption of the RS system. Our glyceraldehyde is also referred to as D-glyceraldehyde, D for dextrorotatory, and the enantiomer as glyceraldehyde is also known as L-glyceraldehyde because of the way the monosaccharides are biosynthesized in the nature. They are named so. Glucose, fructose, and most other naturally occurring monosaccharides have the same chemical configuration as diglyceraldehyde at the chirality center, furthest from the carbonyl group. In the Fischer projections, therefore, most naturally occurring sugars have the carboxyl group at the bottom chirality center pointing to the right. All such compounds are referred to as D sugars like diglyceraldehyde, D-ribose, D-glucose, and D-fructose. All these are examples of the D sugars. In contrast with D sugars, L sugars have an S configuration at the lowest chirality center with the bottom hydroxyl group pointing to the left in Fischer projection. Thus, an L sugar is the mirror image of corresponding D sugar and has the opposite configuration from the D sugar at our chirality centers. D and L notations have no relation to the direction in which a given sugar rotates plain polarized light. A D sugar can be either dextrorotatory or levorotatory. Actually, L for levorotatory. The prefix D only indicates that the hydroxyl group at the lowest chirality center has R stereochemistry and points to the right when the molecule is drawn in a standard Fischer projection. Note also that DL system of carbohydrate nomenclature describe the configuration at only one chirality center, but doesn't say something about the configuration of other chirality center that may be present in many carbohydrates. So moving towards, I'll discuss configuration of aldoses. Aldotetroses are four carbon sugars with two chirality centers. Thus, there are 2,2,5,4 possible stereo isomeric aldotetroses are two DL pairs of enantiomers named erythros and thros. Aldopentoses have three chirality centers and a total of 2, 3, 5, 8 possible stereoisomers or four DL pairs of the enantiomers. So these four pairs are called ribose, ribose, xylose, and lexose. All except lexose occur widely in nature. D-ribose is an important constituent of RNA, ribonucleic acid. L-arabinose is found in many plants, and D-xylose is found in wood. Endohexoses have four chirality centers and a total of 2, 4, 5, 1, 6 possible stereoisomers or 8 DL pairs of enantiomers. The names of the 8 are allose, altrose, glucose, and so on. Only D-glucose from starch and cellulose and D-galactose from gums and fruit pectins are widely distributed in the nature. Actually, these are their extraction sources. D-mannose and D-talose also occur naturally, but in lesser abundance than others. Fisher projections of the 4, 5, and 6 carbon dealdoses to dealdotetroses by inserting a new chirality center just below the aldehyde carbon. Each of the two aldotetroses then leads to two D-aldopentoses, and each of the four D-aldopentoses leads to two aldohexoses. In addition, each of the D-aldoses. So it is their configuration. D-glyceraldehyde converts into D-erythrose, D-therose, and they further configured into D-ribose, D-arabinose, D-xylose, D-lexose, and further configuration when occurs of D-ribose, it converts into D-allose, D-altrose, and D-arabinose converts into D-glucose, D-ammonose. D-xylose converts into D-glucose, D-iodose, and this converts into D-galactose and D-tallose. Then anomers. Aldehydes and ketones undergo a rapid and reversible nucleophilic addition reaction with alcohols to form hemiacetals. Like in this reaction, an aldehyde converts into in hemiacetal. If the carbonyl and the hydroxyl groups are in same molecule, an intramolecular nucleophilic addition can take place, leading to the formation of a cyclic hemiacetal. 5 and 6 membered cyclic hemiacetals are relatively strain-free and particularly stable, and many carbohydrates therefore exist in an equilibrium between open chain and cyclic forms. Glucose, for instance, exists in aqueous solution primarily in the six-membered pyranose form, resulting from intramolecular nucleophilic addition of the hydroxyl group at carbon number 5 to the carbon number 1 carbonyl group. The word pyranose is derived from pyron, the name of the unsaturated six-membered cyclic ether. Like cyclohexane rings, pyranose rings have a gel-like geometry with axial and equatorial substituents. By convention, the rings are usually drawn by placing the hemiacetal oxygen atom at the right jaw as shown uh, in the above figure, which we have discussed in the stereochemistry, a configuration of aldosis. Note that an hydroxyl group on the right in a Fisher projection is on the bottom face of the pyranose ring, and an hydroxyl group on the left in a Fisher projection is on the top face of the ring. For D sugars, the terminal CH2OH group is on the top of the ring, whereas for L sugar, this group is on the bottom, where an open chain monosaccharide cyclizes to pyranose form. A new chirality center is generated at the former carbonyl carbon. Two diastereomers called anomers are produced. The hemiacetal carbon atom is referred to as the anomeric center. For example, glucose cyclizes reversibly in aqueous solution to a 37 to 63 ratio mixture of two anomers, and this ratio is also present in this reaction, like when alpha D glucopyranose converts into beta D glycopyranose, then the percentage is 37.3%, ratio 62.6%. So the compound with its newly generated hydroxyl group at carbon number 1 cis 2 hydroxyl group at the lowest chirality center in a Fisher projection is called the anomer. Its full name is a D-glucopyranose. The compound with its newly generated hydroxyl group turns to the hydroxyl at the lowest chirality center is called its anomer. Its full name is beta D-glucopyranose. Note that in beta D glucopyranose, all the substituents on the ring are equatorial. So it was the brief presentation on the stereochemistry of carbohydrates. Thank you.